Okay, so last time we presented was about 8 months ago. So this presentation will cover slightly different topics and also provide an update while trying to make things interesting. So last year we published this paper called the Characteristic Direction Method. And it's a method to prioritize differentially expressed genes for microarray or RNA-seq experiments. So in this paper, we showed that this method is much more sensitive in calling the right differentially expressed genes compared with all popular existing methods. And we compare the characteristic direction method to fold change as an alternative, the t-test, SAM, or Lima. So here the fold change is shown in green, the t-test is red, SAM is yellow, and then the Lima is in the dashed blue line. And one of the benchmarking strategy that we used is to find publications where transcription factors were either knocked out or knocked down, and then gene expression was measured before and after the removal of the factor. And then we also looked for publications, ChIP-seq publication or ChIP-ChIP publication, where the binding sites for the transcription factors were independently identified. So we did not care so much if the experiments were done in the same cell type, and we also combined human and mouse studies to have a high coverage. And we found about 73 transcription factors that had data from ChIP-seq and ChIP-ChIP, as well as knockdown, removal, and then followed by expression. So then this benchmark was useful for asking whether which of those methods that rank differentially expressed genes can perform better in its ability to identify the putative target from the ChIP-seq studies. And as you can see, the characteristic direction method, the method that we published, beats those other methods by a lot. And the reason we think that's the case is because our method works very different. It's a multivariate methods, and all those other methods are univariate. Basically, they compare each gene to itself under the two conditions without considering the expression of the correlation structure of expression across different genes. So now we have very powerful methods in our hand to analyze gene expression data. So one of the things that we've done is we reanalyzed all of the links L1000 data using this characteristic direction method. And then we wanted to see whether our method is better in extracting more knowledge out of the links L1000 data. So just to remind you, the links L1000 data is a method to, the L1000 is a method to profile genome-wide gene expression. It's developed at the Broad Institute, and the experiments are carried out by the Connectivity Map Group, which is a part of one of the links data generation centers. They measure the expression of 1,000 genes and then impute the rest. So the gene expression of the 1,000 genes, or those 1,000 genes were selected in a way that they will be most informative or most orthogonal genes that can tell you the expression of the rest. And the expression is measured by a Luminex bead technology. It's similar to microarrays, and it's very cost effective. So they've done over 1 million experiments, which is almost the same size as the number of experiments that are available on GEO. And they profiled, they used over 20,000 small molecules, including all of the FDA-approved drugs and many other small molecules of interest. And they applied those to a core of 10 cancer cell lines and measure expression after treatment of those cells with different concentration and measure expression at different time points. So what we've done, we tried to ask a pretty ambitious question, and that was like, can we find gene expression similarity for small molecules and compare that to structural similarity? And people already know that just because two small molecules are similar in structure, that does not mean that they're going to induce the same expression signatures. But what we found is that if you use the method that is currently used by the Broad to call signatures, and it's called the moderated z-score, and this is here stands at MZ, you do not find any relationship between chemical structure and expression. However, if you use this characteristic direction method that I mentioned, you get some signal, and a pretty strong signal between expression signatures, correlation, and structural correlation. And one of the other things that this can be done, it can be used to benchmark the various methods that are used to define structural similarity. So we use two methods to define structural similarity, and for that you have to convert the SMILE string of the drug structure into a binary string and then define some measure of distance, and this is done by, there are various methods to do it, and we tried both, two of them, which one is called MAX and the other one is ECFP4, and you can see that the ECFP4, it's much better at defining structural similarity between drugs because it confirms more expression similarity between drugs. So some of those BD2K considerations, so that characteristic direction method is clearly doing something very good in identifying and prioritizing differentially expressed genes. However, it hasn't been cited that much or used by many. And so there's sort of like some BD2K considerations here to, and one is based on the Twitter post by Andrew that mentioned how this program David, which is used to perform enrichment analysis, was cited over 2,000 times in 2014, while they haven't updated their annotations in over 5 years. So in general, first, our ability to create those benchmarks that can potentially be used to evaluate pipelines, computational pipelines, I think it's a very key thing that will be appreciated more and more in this BD2K arena. And in order to do that, you need data from two independent resources. For example, here we use ChIP-seq data for transcription factors and gene expression for transcription factor removal. And also we show how like drug structure similarity can be used to look at drug-induced expression similarity. So those type of independent resources, you can freeze one, make it a silver standard, and then evaluate your methods by comparing how processing the data from one data set is confirming the agreement with these other independent data sets. And then we want to ask like how, in general, tools that are potentially better are not being adopted. So we're starting to see this effect where tools that are marketed better or people are sort of like stick with are becoming more adopted. Hi. So one of the things that we realize now that we have this characteristic direction method, all of those studies that are on GEO potentially have a lot of knowledge that still hasn't been extracted, and we were wondering like what can we do about potentially processing or extracting more knowledge from those studies on GEO now that we have this tool. And what we've done, we developed this relatively easy to implement tool called GEO to Enricher, which is a Chrome extension. And you can download and install this Chrome extension from the Google Play Store by just clicking on one button, and also all the updates are happening dynamically. So once you install it, you will get the new versions automatically. So when you install this tool, nothing really happens until you go to GEO. And yeah, so recently we had some collaboration, or we were working with a group from the University of Manchester who is studying the glucocorticoid receptor. And in their experiments, they transfected HEC293 cells with glucocorticoid receptors and stimulate those cells with dexamethasone. And then they measure expression before and after using the drug. And there are already similar studies on GEO. So in the past weekend, I was checking the consistency of these studies by using this tool, the GEO to Enricher tool, and I typed in DEX in the search bar, and here I'm showing how I processed the third study. So once you click on that third study, you see that there is a new button that is added to the gene datasets page. And in this particular study, the authors wanted to identify the effects of applying DEX to airway smooth muscle cells, because DEX is one of the uses of it. It's an FDA-approved drug that is a steroid that is used to treat asthma. So once you select this study, you see this button, and then you can click on that button to invoke the effects of the Chrome extension. So Chrome extensions, they basically hijack existing pages, and they can now be used to control the JavaScript and the HTML of the page, and potentially can use to enhance the functionality of web pages. So here we're trying to enhance the functionality of GEO. We are still using the GEO functionality for the user to identify the groups that we want to compare. So here I selected 4 untreated cells, 4 samples from untreated cells, and the 3 samples from the 4-hour stimulation with DEX. After that, once you click on that extract signatures for GEO, you get this window that shows you what you selected and allow you to enter additional metadata and also some metadata tags. So once you finish entering the data, you click Extract Gene Lists, and at that time our request is sent to our server. We download those sample files, we process them with this characteristic direction method, and then we return a results page that looks like this. So that result page has a permanent link that you can always come back to, and has those buttons that some of them are to visualize the data, and some of them are for downstream analysis. That you can also download the results, and I'll show you some of the downstream analysis. But before that, one of the things that we recently implemented is a 3D plot. So here you can see that the control samples, the 4 control samples, are nicely separated from the treatment samples. Next, I clicked on this green icon, and that icon takes me to a tool that we developed called L1000 CDS Square, which stands for L1000 Characteristic Direction Signature Search, which is a search engine that we developed by processing all of or a big chunk of the data from the L1000 with this characteristic direction method. So this is the results from the search for additional drugs, or L1000 drugs, that can mimic the signature that we observed from that study in GEO. And I think these results are really awesome. And what the reason this is like very exciting is that you can see DEX twice in the top 10. And this means a lot. It means that first, the GEO study is a good study. It means that the L1000 data is a good data set. It means that our characteristic direction methods work. And it means that the DEX effect can be distinguished across cell types, concentrations, and experimental platforms. It means that all those other drugs that are in the top 10 list are working similarly to DEX. And a quick Google search of those drugs show that indeed they are doing similar things, and some of them are known FDA-approved drugs. For example, ketorolac is an NSAID, a non-steroidal anti-inflammatory. Perhaps it's a better alternative to using DEX in some cases, and perhaps it may be actually a steroid, but people don't know that yet. They think it works as an NSAID, but it could be that it's working through the GR receptors. This drug, thalidomide, is a drug that is already used in combination with DEX to treat multiple myeloma. And it was also used to treat leprosy in the 50s. Another drug that is up there in the 9th position is a drug that is very similar to DEX, and it's already used to treat allergy. So this is a steroid medicine that. So what also, if you look at this broad compound, this is a compound that is a part of the L1000 data that hasn't been used, but it's sort of like in a good company here of drugs that are doing similar things. So this is a great candidate for further exploration and having potential to have similar effects as those potentially useful drugs. The other thing that this tool provides is an ability to understand the mechanisms of the drugs and how those genes that are differentially expressed by the drug what are they doing in their pathways. So this is take you to this tool called Enricher, which I mentioned before, like in my presentation in February. And Enricher is a gene set enrichment analysis tool that we also developed, and it shows how the two tissues that have most overlap with the upregulated genes in DEX are the two tissues that we already know that DEX is very useful for, in general, for treatment those diseases. So one is lung, and the other one is those plasma cells. We also find when we do enrichment that there is high overlap of the upregulated gene with the GR receptor. So NR3C1 is a synonym, or it's the gene symbol for GR alpha. So we get confirmation that DEX indeed activate targets of GR alpha, and again we see consistency across experiments and platforms and types of experiments. So here are some more BD2K considerations. So one of the things that we believe, or we want you to pay attention to, is that once, with BD2K, we want to organize all those data sets and serve them in a useful way for reusability. But I think what's key here is also to have those buttons that link you outside to downstream analysis. either data visualization or integration with prior knowledge. So the tool GeoEnricher, the results page, gives you those small icons that takes you to this downstream analysis. Since February, I looked at the presentation from last time, and when I took a screenshot of the Enricher tool, at that time there were 127,000 lists uploaded to the tool, and since then, this weekend, we had 442,000 lists uploaded. So this sort of like show you how this tool is becoming really popular, and as a aggregator of gene lists from the community, it's also can become a resource for collective knowledge. And we hope to have like within a year to break the 1 million gene list uploaded to this tool. The other sort of like good news is that with the example of GR, and we also looked at interferon alpha and other examples, we do get reproducibility of gene expression data from different assay labs, cell types. And this is good news because people are talking so much about how data is not reproducible. But if you look at it in a statistical way, and you use the right methods to process the data, or better methods, then you will start seeing consistency. And it's sort of like looking at the half full part of the glass of this issue of reproducibility. And we see this recovery of the GR, transcription factor from ENCODE, and the ranking of DEX in the top 10 for when we query it against the L1000 dataset. So one thing that we also did in the last 6 months, which was a lot of work, was to run this Coursera BD2K course on Coursera. And within this course, we try to recruit students to use the GeoEnricher tool to extract expression signatures from GEO. By the way, this course is still open and running, and it's not too late to sign up. So for the course, we added, for this course, we added 4 new microtasks. And one of them is perturbations of MCF7 cells, which are breast cancer cell lines, comparing young versus old tissues, looking at endogenous ligand perturbations, and looking at signatures where viruses, bacteria, or other microbes were challenging human or mouse cells, and measuring expression before and after. And the numbering, so the top 3 listed microtasks are the microtasks we run for the course that we started, that was running from January to March this year. And those are single gene perturbation, drug toxin perturbation, and disease versus normal signatures. Those have more entries, and those are a little bit more broad categories. But I think it was pretty successful to have, even though there were not a lot of participants, there's some very dedicated participants. Okay, let's continue. I wonder what that symbolizes. Anyway, so next, I wanted to just give you an update on this project called Harmonizome, which also we made a lot of progress with. And last time I just mentioned it. And now we have already developed this website that provides access to this. So this is just a reminder that we have been processing and extracting gene sets and attribute tables where the genes are the rows, and then the functions or various properties of genes are the columns from many public resources. We have processed 66 resources, and in this page you can see a list of them. And we have those data sets, like processed data sets, that provide access to knowledge about genes, and those can also be used for machine learning to impute knowledge about genes. And the interface to the harmonisome is a search engine trying to make it very simple. But most of the people that use this site, which was only published a month ago, they find information for it on Google. So about a month ago, we actually put Google Analytics on this site, and we found that it's highly accessed. So 4,000 users in 1 month before it was published or anyone ever found about it. It was pretty surprising. Yes, I'm almost finished. So this is then looking a little bit deeper. It shows that all of those users are coming from Google by searching gene names. We also developed this Harmonisome mobile app that allow you to find knowledge about genes and proteins. So the mobile app is available on the App Store, the iPhone, and Google Play. And basically, it's just it allows you to enter a gene name and then select a category and then explore knowledge based on all of those resources that we integrated. And this is the interface. And I'm going to sum up with 2 more nuggets of things to consider. And one of them is the phone app that we developed did not take more than 2 weeks to actually put together. And right now, it's becoming very easy to develop those phone apps. There is only 2 major platforms, and Mike McDermott, who developed the tool, used this React Native platform that was developed by Facebook to create this app. So we are just letting people sort of like think about making their web tools, mobile, without too much effort. And the other thing is the Google showing us that people are searching for gene names. So I will sum up here, and I'll take any questions if there is time.
The sobering thing that I thought you said was your concern about the time it takes to diffuse good ideas, and your discussion of Andrew's tweet was reminiscent of the often quoted 17 years it takes for clinical trials to have their results disseminated. Are we in the same business with software, and do you have any suggestions on what we might do? It seems like indexing is never going to be good enough.
Yeah, I think that the idea of benchmarking and setting up, standard benchmarks across that are sort of like official benchmarks that people can test their methods, especially for RNA-seq processing. I think it's very important. And other type of genomic sequencing data, there should be some benchmarks that will tell the community what would be the best practices to apply the best methods, and there should be some reward and competition for computational biologists to develop those tools, and that could potentially accelerate this adoption of the right tools.
In a couple of weeks, you've all seen the draft agenda, and I think you've all registered, so thank you very much. And hopefully there are a lot of abstracts that have come in for the poster session, you'll recall that the center's got one of the sweet spots on our agenda. It's right after Phil talks and right before Francis Collins on the first morning. So that's all the good news. The bad news is that you have from 9:20 until 10:30 in order for each of the 12 centers, as well as the Golub UO1 Award, to be able to make a short presentation each. And as we're currently planning it, we're thinking your single most impactful finding to date. One of the ways we were thinking of providing some guidance is no more than 2 slides, with the first possibly addressing scientific outcomes so far, and the second to address developments that could impact the rest of BD2K. But what we're really looking for is, it's hopeless. You'll never be able to touch on everything that your center is doing or will do. So think of this as your elevator speech, and this is your moment to say, hey, right now, this is the most impactful thing we have done or we are doing and why it's impactful.
Please, we have a third slide that tells people before anything else what our center does.
Sure, you could do that. What I was about to say, however, which makes a lot of sense, is this. Francis Collins needs to start talking at 10:30, and he must leave at 10:45 because he has many back-to-back appointments. And my concern is that all of the awards have a chance to talk. So I wanted to put this out there and lay it before you and say, you know that things always go slightly haywire, and I know this sounds horrible, but do you think it would be possible to aim for like the 3-minute side of 5 minutes rather than the 7-minute side? Just because I'm afraid that we're coming up to 10:30 and only 11 centers have presented. So we either have to cut out 3 centers, or we have to cut out the NIH director, which sounds like a bad plan. So I wanted to put that out there and get everybody's input.
Can you hear me?
Not really.
Okay, I pick up my phone. Okay, so in order to make this happen, you have to collect the slides ahead of time. And
yes, and the coordinating center is doing that.
Yeah, so someone need to be putting the traffic light when the time is coming short to, to warn the PI to stop, prepare to stop.
Yes, we can definitely do that. That is not a problem, but having been at a number of things where once or twice I've had people go out of their way to ignore the traffic light and the person trying to drag them off the stage.
Use the hook. We drag the PI.
We also need the agreement from everybody to be responsive to make sure we stay on track. And I'd rather bring this up with you all ahead of time and do this cooperatively with you rather than just lay down the law.
Okay.
One suggestion, you could adopt the Ignite format where slides are assembled and auto-advance on a timer.
Oh, we've never done that. That's a clever idea. That might make it a little challenging for people with multiple slides and might be a reason to... What do people think? Because there are a lot of bonuses to that approach, but if you want to spend more time on one slide than another, it might be problematic.
Yeah, so Jenny, this is Paul Thompson here from the Enigma Center. I think that sounds like a really fun session. It'd be fabulous, and so long as you have an MC that can kind of make sure that people don't overrun, it's fine. My only question is that if people have some kind of burning question during these, it could kind of derail your plan to have it all complete within the hour. So I mean, I guess there's pluses and minuses either way. Maybe you just want to tell people that give them the the goals of it. Don't ask questions. I mean, do you have any thoughts on that? Because I'm sure.
Yeah, my thoughts are we can only take questions at the very end, and if there's time. And we should heavily plug the poster session, because that is where more engaged and in-depth and meaningful discussion can happen.
Yeah, I like that a lot. I mean, I would agree with Mark. Maybe just let people have 3 slides and wrestle them off the stage after 3 minutes or 4 minutes or whatever you guys decide. But it sounded like a lot of fun, and if people go into it knowing that it's time limited, they'll behave, I think.
Jenica, I know from personal experience you're really good at getting people off the stage.
Art Toga is also most excellent at it. I just want to give him the hats off.
Wait a minute here. I don't want to get credit for that.
So it sounds like what we have some consensus coming to is up to 3 slides, and Soninka will work to fine-tune the timing, but I'd say for now, assume you'll have 4 minutes, because I'm just going to assume there's going to be something at the start and then something at the end that will eat up at least a few minutes. Does that sound reasonable?
This is John Gravio. I would recommend that you actually tell everybody it will auto-advance at 1 minute per slide, and if they want 2 minutes on the slide, they put 2 in and they get 4 slides. They can put the same slide in twice. And you have 1 minute that is for the change of, or 30 seconds for the change, and make that a slide, and it just keeps going. And everybody will get it right away that they have.
Oh my God, that is brilliant. What do people think? Do you want to be self-regulating, or do you want to be helped by technology?
That's a little draconian. I think people should be responsible.
Hear, hear.
All right. So although I was entirely charmed by that proposal, nobody else was. So we'll go with 4 minutes and a strong border collie to keep people on time. All right. The other thing I wanted to let you all know is that over the next couple weeks, as we get closer to the all-hands meeting, if you have any questions, concerns, anything along those lines, don't ask me, ask Soninka, because I will be on vacation on a road trip driving to Arizona.
From DC?
Yeah.
That's a road trip, okay.
Yeah.
Wow.
Because people probably have not completely made their travel plans, when we spoke last month, there was some discussion of some peri-event events. And is that still something that we should be planning for, or are things still uncertain?
All right, the peri-event events, the one that I do know of is an interest in organizing some sort of informal get-together on Wednesday evening for the people who happen to be around. I don't know more details than that, so I would say make the travel arrangements that make sense to you. And if you're here and that comes to fruition, yay. We are starting at 9 a.m., not 8 a.m. East Coast time, but it will still mean that folks will have to get in general the night before, no matter where you're coming from. Even if you're coming from Boston, it would be challenging to fly in and get all the way through Natcher. Oh, reminder, Soninka is working with our contractor to help ensure that there are shuttles and other ways of getting you all onto campus. We did listen, and we're starting it a little bit later to make sure there's time. But and we are working with NIH security to try to facilitate the getting onto campus screening and all that. But someone else had a big meeting just a few weeks ago, and it took me a long time to get through security. So make sure you and the various folks you're bringing don't try to waltz onto campus at 8:45. Give yourself a bit more leeway. I heard there were about 350 people, of which about 250 are various BD2K funded investigators, and the other 100 are NIH staff. And a good thing, because we're very much looking forward to more people at NIH really knowing all the amazing things you guys and the rest of the awardees are doing, so they can go back to their ICs and proselytize.
Proselytization.
Excellent.
Okay, can we move on to the next agenda item, or are there more questions about the all-hands meeting? Okay, Alex and Brian, how fast can you talk?
I can talk really quickly. There are 3 working groups at this point: metadata, which you are all well aware of. We've actually created a working group around APIs, and we also now have a interest in a clinical informatics working group with several people from different centers in both the API and the clinical informatics group. The intent is that certainly the working groups will have part of the breakout sessions to meet at the all-hands meeting, and hopefully we can get people together there. Finally, we are still working on getting the electronic infrastructure up to support all 3 of the working groups. We actually have an initial prototype, but I haven't released it formally as of yet because I'm not quite happy with its presentation, but it should be good to go by the all-hands meeting.
Centers Coordination Center. I'll be really quick here. We are going to be collecting slides for you guys to demonstrate your single most impactful finding during the all-hands meeting, as we mentioned. So I'll be sending out an email soon to collect slides. And also, if it's more convenient, there'll be a link to the website where you'll be able to upload your slides. And we're going to assemble those into a single presentation so that hopefully they'll be able to go by very, very quickly.
Posted now on the GoToMeeting. There are a number of things we could do as all of us together for that 90 minutes. So whatever your suggestions might be, send it to us. Currently, I have about 3 different things I thought we could be doing, and they don't have to be all necessarily in the agenda draft. One is what would be the most important milestones that you consider for your center to accomplish in the coming year. And the next I thought is important or interesting, stimulated by the discussion we have had at the Palm Spring meeting organized by Art, is looking at what specific projects that all of us could collaboratively work on. These would be projects that couldn't be accomplished by any given single effort from one center. So that's something we could talk about in that 90 minutes of time before we report back to BD2K overall. The last thing is sort of BD2K Centers Coordination Center milestone thing, is we really would like to work with your center to promote your accomplishments and broaden the impact of what you have achieved. And that requires your help on letting us know what are the specific things you would like us to do to support you. One thing is each center has our own specific scientific organizations, whether it be 2 or 4 or 5 of them that we are closely associated with, and we could help you to support your workshops or symposiums or whatever you wish to have in these scientific organizations, and helping you build platforms for dissemination of the tools BD2K will generate. Just a thought, and any other recommendations you have, please send it to us. Next one. I'm done.
Its annual meeting. that Phil attended. And so in this meeting, basically all the investigators and students and postdocs, they all came together, and we discussed the previous year's findings with the posters and demos presented by all the students and postdocs of the center. So I'm not sure if there is any more update that you are looking for, so I've just quickly summarized what we did in the annual meeting.
That's great. Henning and Andrew, tell us about HUPO.
Okay, so this is Henning. We organized the Bioinformatics Hub as a satellite to the Human Proteome Organization conference in Vancouver. And we had one room as a meeting point for bioinformatics interested people for the entire time during the meeting, which was a major contribution by the organizers. And the meeting went really well. There were between 30 and, well, 5 at low times, 30 at high times people in the meeting. Overall, more than 50 participants. And it was a great communication point between the experimentalists and the bioinformatics people. And the main point for people to take away is having one point as a meeting point for during a larger conference is a great thing. But of course, you need to organize this.
Great, thanks so much. Art, tell us about Palm Springs.
Oh, it was a great meeting. It was focused on the West Coast contingency of BD2K awardees, and we put together an agenda that really allowed for collaborations to emerge and to find common working opportunities between us. So we did a little bit of coverage of what we do and what opportunities there might be for us to link these efforts in a mutually beneficial way. So we came away with a number of items which are being written and described, and we can distribute those. In fact, they'll be posted on the CC site, the BD2K CC site, for everybody to look at. This is in no way meant to be exclusive by any means, but it was just kind of geographically useful for us to meet here. And that's about it, basically. I think it was a good success. It was the right period of time. It was basically 1.5 days, and I think everybody enjoyed themselves.
Fantastic. And I guess a number of you were involved in a meeting called Big Data Challenges in Life Sciences.
I was? How did I do?
Well, someone has to tell us about it. Mark, are you going to speak, or?
Sure.
No.
I can.
Someone speak.
So this was a symposium organized by the Institute of Bioinformatics at the University of Georgia on October 12th. And the BD2K consortium was very well represented among 6 keynote speakers. 4 of them were BD2K Center members, along with Phil Bourne. So Phil gave the first keynote, Pei Ping gave one, Greg Cooper, and I all gave keynote addresses there.
You can understand basically where we are presenting, what kind of an impact we're having, and I'm wondering in what way we engage the coordinating center to help promote these meetings and to summarize our presentations there. That's more of a rhetorical question, because I'm not sure what the answer is.
We collected feedback forms, and these will be available through the center.
This is Pei. We also welcome any kind of recommendations, formats you may have. I recently was also at a meeting in China together with Henning that was co-organized by Nature Publishing Group with one of the omics groups in Beijing. Actually, 7 of the Nature Publishing Group editors or senior associate editors showed up. And there are discussions as we have with raising our profile in the journals in different aspects of the scientific discipline. It does require joint efforts from all the centers. It's not something anyone could do it alone. There is also a specialty journal in Cell called Cell Systems that are also very interested in BD2K activities, and I'd like us to welcome their participation in the next set of events that all of our centers are going to have, either at NIH or other centers and symposiums with any joint efforts.
Sounds good.
NIH, we have some insanely long lead times to get approval for travel sometimes. So if you could let your program officers know when you either are intending to have a meeting or things like that, as soon as humanly possible, that would be greatly appreciated. Just because we sometimes need to know at least 6 months ahead of time. I know you don't always schedule things that far ahead, and that's fine, but when you do have them scheduled far ahead, please let us know so we can put that in our travel approvals.